Have you ever really stopped to think about what's happening inside your head right now at a microscopic level? It's not just a few sparks, it's a communications network way more complex than the entire global internet, all firing away inside your skull. So how does it actually work? Let's follow a single message on its incredible journey deep inside one of your brain cells. It really all comes down to this one central puzzle. You have a single molecule, a neurotransmitter, that shows up at the surface of a neuron. Now, it can't get in. It's stuck outside. Yet somehow it has to deliver a message that can completely change how that cell works. So how in the world does it pull that off?
Well, the answer is just, it's amazing. It's not a single event. That one little signal from the outside kicks off a lightning-fast cascade, a kind of molecular relay race that doesn't just pass the message along, it amplifies it into a powerful command.
Okay, so let's start at the very beginning, at the cell's front door. Our messenger, the neurotransmitter, has arrived. But the cell membrane is basically a wall. So the message is stuck on the outside, and all the machinery it needs to control is on the inside. That is the fundamental problem the neuron has to solve, every single time.
Now, there's a scientific name for this whole magic trick, and it's called signal transduction. All it really means is taking a signal from outside the cell and translating it into an action inside the cell. And look, understanding this is absolutely key because it's not just how our brains work moment to moment. It's how we learn, how we adapt, and it's the exact mechanism that most psychiatric medications are designed to tap into. So a neuron actually has a few tricks up its sleeve for getting these messages. 4 main ways, in fact. Some are incredibly fast, like a gate just swings open, and that's it. But others are way more sophisticated. They kick off a whole chain reaction involving things called G proteins and second messengers, and it's this more complex system that we're going to focus on, because that's where the really profound, long-lasting changes in the brain begin. To get that message across the border, the neurotransmitter first has to find the right person to talk to, a specific receptor on the cell surface. You can think of these receptors like highly specialized doormen. They are incredibly picky, and each one is designed to recognize only one very specific type of message. And there are really 2 main kinds of these doorways. First, you've got your ligand-gated ion channels. These are like a simple doorbell. You press it, and bam, the door swings open instantly. Ions rush in, it's a direct, super fast electrical response. But then, you have the G protein-coupled receptors. These are the smart home hubs. You press this button, and it doesn't just open the door. Oh no, it kicks off an entire sophisticated program of events inside the house. And here is the absolutely crucial point. While those fast-acting channels are vital for things like reflexes, the vast majority of neurotransmitters, and really importantly, most drugs that affect our brains, they all target these more complex smart hubs. They just allow for a much richer, more powerful response. So what happens after that smart hub gets the signal? All right, so once that smart hub receptor is activated, this amazing internal process just explodes into action. And the goal is no longer just to pass on the message, the goal is to amplify it, to turn that one single tap on the shoulder at the surface into a powerful, thundering storm of signals all throughout the cell. And this whole show is powered by something called a G protein. Think of it like a little machine, just waiting to be turned on. When it's inactive, it's holding on to a low-energy molecule. But when the receptor gets that signal, it yanks that one out and slams in a high-energy one. That simple swap is the on switch. The G protein springs to life, its parts break apart, and they race off to activate other bits of machinery in the cell. And get this, it has its own internal timer that automatically switches it off, reassembling the whole thing so it's ready for the next signal. It's just so elegant.
So what are those little pieces of the G protein racing off to do? Well, they're starting a process that relies on what the source material calls the major molecular currency of the cell's internal pathways. And that currency is something called protein phosphorylation. This is the key action everything has been building towards.
And here is where the true power of this whole system just comes roaring to life. The cell doesn't just relay the signal one-to-one. No way! It multiplies it massively at every single step of the way. This is how one tiny little event on the outside can have such a gigantic impact on the inside. It all starts right here, with just one single neurotransmitter molecule binding to one single receptor. But that one single activation triggers the creation of hundreds of what are called second messenger molecules inside the cell. A really common one is called CAMP. So all of a sudden, that initial whisper from the outside has become a powerful chorus on the inside. And the amplification doesn't stop there, not even close. Those hundreds of second messengers then fan out and activate thousands of proteins. Just think about that scale. We went from 1 to hundreds to thousands. The initial signal has now been magnified exponentially, creating this powerful cell-wide response, all from one tiny trigger. Okay, so we have this massively amplified internal signal. We've got a storm of messages flying around inside the cell. But so what? What does the cell actually do? How does it translate all this noise into a meaningful change in what it's doing? It all comes back to that molecular currency we mentioned, protein phosphorylation. If you only remember one term from this, this is it. Think of it as the cell's universal on-off switch. An enzyme comes along, it slaps a little charged phosphate group onto a target protein, and that one tiny addition instantly changes the protein's shape and its function, basically telling it to either get to work or to stop what it's doing. Now, the enzymes that do the actual switching, the ones flipping the switches, are called protein kinases. You can think of them as different teams of workers. The second messengers we talked about, like CAMP, they don't do the physical work themselves. They're like the managers shouting the orders. Their job is to activate a specific team of kinases, and these kinases are the real workforce, spreading out through the cell to flip the on switches on thousands of other proteins. Of course, a switch that's permanently stuck in the on position isn't very useful, right? That's why the cell has an equally important system of off switches. These are called protein phosphatases, and their entire job is to come along and remove those phosphate groups, resetting the proteins back to their original state. It ensures the whole signal is temporary and very precisely controlled. So far, pretty much everything we've talked about happens in milliseconds, maybe a few seconds. But here's the most profound part of this whole story. This rapid, temporary signaling cascade can be used to create very permanent, long-term changes in the brain. This is literally how we learn and how we remember. The ultimate destination for some of these signaling pathways isn't just a nearby machine. The message can travel all the way from the outer membrane, across the entire cell, and right into the central command center, the nucleus. That's where the DNA, the cell's master blueprint, is stored. And once that signal gets inside the nucleus, it can act as a master regulator. It can activate proteins that physically unpack the DNA, allowing specific genes to be read. It can change which genes are turned on or off. This fundamentally rewrites the neuron's day-to-day operating instructions, creating physical, lasting changes that form the very basis of long-term memory and even the therapeutic effects of many long-term drug treatments. So just think about the journey we just took. From a single molecule knocking at the door to a cascade that amplifies its message thousands of times, all the way to rewriting the cell's genetic master plan. This process, signal transduction, is the fundamental language of the brain. It's how a fleeting experience can become a permanent part of who you are. And the ultimate goal of neuroscience, really, is to become fluent in this language, so that we can finally learn to write our own messages, and maybe, just maybe, correct the cellular miscommunications that lie at the very heart of so many brain disorders.